Hello dear viewers, I hope you all will be fine and doing well. Dear friends, dear viewers, in this lecture video I will discuss an amazing topic of types of structural changes which occurs in the chromosome. In the previous lecture videos I had discussed the different types of monosomy, polysomy and trisomy. I hope you will like my videos. I hope you have watched my videos. If you are watching my videos and watching my YouTube channel Authentic, then here I kindly request you to please subscribe to my YouTube channel and like my video, share my video, and do support my YouTube channel. Dear friends, dear viewers, our today video will be types of structural changes that occurs in the chromosomal structure. So the first, there are four types of the structural changes in the chromosome. The first one is deletion, the second one is duplication, the third one is translocation, and the fourth one is inversion. So here we will start it with a deletion. So deletion or deficiency, it refers to the loss of portion of segment from a chromosome. Based on the location of loss of the genes, deletions are divided into two types. The first one is the terminal deletion, while the second one is interstitial deletion. Terminal deletions is the loss of a portion from terminal ends of a chromosome, which is known as terminal deletion. It may have two types: heterozygous, in which the deficiency occurs in only one chromosome of pair, and homozygous, in which the deficiency occurs in both chromosomes of a pair. Interstitial deletion is basically it is a loss of intermediate portion of a chromosome or between telomeres or centromere. It is also known as intercalary or interstitial deletion. The deleted portion comes out and remaining segments are reunited. If you talk about the effects of deletion, so in the effects of deletion, here we have the small deletion, which we can also call it heterozygous deletion, while another one is large deletion, which is a homozygous deletion, and it is a lethal for an individual. So homozygous deletion is not lethal, is not dangerous for an individual, while large deletion, which is also called homozygous deletion, which is it is lethal for an individual. Crossing over is also suppressed in presence of deletion due to loss of corresponding portion in homologous chromosomes. Deletion also affects phenotype due to deletion in short arm of chromosome, which causes Cre-du-Chat syndrome and males. While the significance is the deletion are important for creating variability by chromosomal mutations and used as a identifying tool for gene mapping. While in the second one is the duplication, the second structural change in the chromosome is duplication. So it refers to the presence of a segment twice in the same chromosome. It is also known as repeat. It is of four types: the tandem, reverse tandem, displaced, and the reverse displaced. So here we will discuss all one by one. So the tandem, in this type of duplication, in the tandem duplication, duplicated segment is present in the same sequence of genes by the original segment of a chromosome. While in the reverse tandem, in this type of duplication, duplicated segment is present in the reverse sequence of the genes by the original segment of a chromosome. The second one is the duplication, so displaced. While in this type of duplication segment is present away from the original segment but on the same arm of the chromosome. While in the reverse displaced, In this type of duplication, duplicated segment is present away from the original segment but on the other arm of the chromosome. If we talk about the effects of duplication, so the first effect of duplication is that it increases the number of genes in the chromosome. Crossing over also suppressed due to the duplication of the chromosome. Charcot-Marie-Tooth disease, it can cause Charcot-Marie-Tooth disease. Duplication do not reduce viability and are harmful than deletions. Duplications leads to addition of some genes which may have an important role in evolution. Translocation, it refers the third type of chromosomal changes is translocation, and translocation refers to the one-way or reciprocal transfer of the segment between non-homologous chromosomes. Translocation differs from the crossing over because it involves non-homologous chromosomes, while crossing over involves non-sister chromatas of homologous chromosomes. Translocation are three types. There are three types of translocations. Simple translocation. In this type of translocation, a segment of a chromosome is transferred and attached to the terminal end of a non-homologous chromosome. In this type of translocation, an intermediate or intercalary segment from one chromosome transferred to the intercalary position of non-homologous chromosome, which is known as shift. Reciprocal translocation. In this type of translocation, one break occurs in each chromosome and occurs mutual exchange of segments between non-homologous chromosome. It is known as reciprocal translocation. These translocations are very common and have great evolutionary significance. Sometimes the reciprocal translocation of non-homologous chromosome are involved in translocation, which is known as multiple translocation. Here we will talk about the Robertsonian translocation. So clinically more important type of translocation is Robertsonian translocation. In this disorder, the long arms of two non-homologous chromosomes fuse at the centromere, forming a single chromosome. Robertsonian translocations are confined to chromosome 13, 14, 15, 21 and 22 because the short arms of these chromosomes are very small and contain no important genes. The short arms are usually lost during subsequent cell division because the carriers of Robertsonian translocations lose no important genetic material. They are unaffected although they have only 45 chromosomes in each cell. Their offspring, however, may have serious monosomies or trisomies. For example, a common Robertsonian translocation involves the fusion of the long arms of chromosome 21 and 14. An offspring who inherits gametes carrying the fused chromosome can receive an extra copy of the long-term chromosome 21 and develop Down syndrome. Robertsonian translocations are responsible for approximately 3% to 5% of Down syndrome cases. Parents who carry a Robertsonian translocation involving chromosome 21 have increased risk for producing multiple offspring with Down syndrome. The effects of translocations are translocation lead to duplication and deletion of gene because gametes of duplication are deficiency are inviable. Crossing over is also suppressed. Translocation may affect phenotype, progeny of individual. Heterozygote with a translocation involving chromosome number 21 can arise as Down syndrome in humans. The fourth one change in the chromosomal structure is inversion. Attributes to the structural change in a chromosome in which in a reverse sequence based on whether a centromere is involved or not involved in the inversion. It is classified into two types: paracentric inversion and pericentric inversion. Paracentric inversion, when the chromosome is not involved in inversion, is known as paracentric inversion, and this type of inversion breaks occurs only in one arm of chromosome. Pericentric inversion, the second type is pericentric inversion, when the centromere is not involved in the inversion, is known as pericentric inversion, and this type of inversion breaks occurs both in each of the two arms of a chromosome. So this is all about the structural changes of chromosome. Thank you so much for watching my YouTube channel Authentic. I hope you will like my video. I hope you will understand my video. If you are watching my YouTube channel and watching my videos till end, then I kindly request you to please do subscribe my YouTube channel Authentic and share it with your friends and keep supporting me. Thank you so much. Take care of yourself. Goodbye.